And today the discussion is going to be on biomarkers for intestinal function. And Jennifer, as you said, we do this with dried blood spot, which makes it very easy to do at home for a client or patient or for anyone in their office. So it really is a wonderful way to be able to do things. So we are discussing the gut this hour, and it has been known traditionally as the portal to health. As Hippocrates said way back, all diseases begin in the gut. And I think he was onto something when he talked about that. So in order to understand what we're going to be discussing, I think we need to say a few words about the pathogenesis or the pathology involved in inflammatory and autoimmune disease. And when one looks at that, there are classically a number of things you look at. One is, what is the person's genetic predisposition? What is it in their genes that will predispose them to a particular problem? Apart from that, you have the environmental triggers, the very many that there are, from toxic chemicals to infections. And in the case that we're going to be describing here, the various types of dietary proteins and peptides which are entering our gut each day. Added to that is we have this wonderful thing, the microbiome, the bacterial world which lives within our gut, and we will be discussing that in more detail in a few moments, especially when it begins to go astray, when it isn't functioning as proper, and that is what's called dysbiosis. So that is important. Those two things together, the environmental triggers and the microbiome, will determine the types of intestinal inflammation that we all deal with in our daily lives and the immune response to that inflammatory process. And that, of course, will then lead to more systemic inflammatory problems and even autoimmune disease. But what has been looked at in the last number of years, which has really been a game changer, is number 4, and that is, how does all that lead to increased intestinal permeability? And we used to call that leaky gut, which is a term which a lot of people are saying is really not what you want to use. It is really intestinal permeability. And when the gut becomes more permeable, it allows what's going on in the gut to not stay just in the gut, but to begin to affect the body as a whole. And that is that interplay between permeability and intestinal inflammation that causes so many of the problems. And so what is the role of biomarkers in all of this? Well, what I'm going to try to do in the next few minutes is talk to you about some of the things that we use in our lab to deal with that. The game changer, the foundational test is zonulin, or the zonulin family peptides, FP, which are known now, quite established, that zonulin modulates or is even able to reverse the process of intestinal permeability. We also want to look at what is going on from an inflammatory standpoint in the gut itself. And for that, we're going to talk a little bit about histamine and its breakdown product, Diamine Oxidase. We're also going to be looking at the efficacy, the importance of looking at various kinds of food sensitivity. And with that, what happens when you begin to get various types of pathogenic changes or dysbiotic behavior, of which Candida, the yeast, is a classic example. It is the prototype of all of that. So that's what we're going to be trying to deal with today. So what are we dealing with when we talk about the intestine? Well, the intestine is really the largest surface of the body. It's about the area of 2 tennis courts. It's vast in its square meter area, and it is the home for something in the order of about 150 trillion with a T bacteria. That is what we know lovingly as the microbiome, and it contains about 15 times the total number of cells that we have in our human body, which is about 10 trillion cells. So it's an incredibly important part of what goes on within the dynamic aspect of our body function. It is continuously exposed to the environment, to various bacteria and dietary antigens on a moment-by-moment basis in our lives. And because of that, and because of the interplay of the outside environment and our body, it is not surprising that the largest number of immune cells, fully 70 or even more % of all lymphoid tissue in the body, is sitting literally intimately attached to the intestinal barrier cells, because it wants to catch things as it happens in the intestine. And to give you an idea of the importance of the gut, 40% or more of the body's energy expenditure happens within the gut. So it tells you something about how important that is. So let's take a look at what is this intestinal permeability stuff that we're talking about. And we're going to discuss zonulin. Intestinal absorption is extremely important, and we do have a pretty good handle on how things go in that area. The intestinal mucosa has a very paradoxical role, because on the one hand, it's absorbing nutrients on a moment-by-moment basis, but at the same time, it needs to be able to exclude toxins and various types of large or macromolecules which it can't absorb properly. So when you get a breach of that mucosal barrier, it can lead to a number of things. One of them is malabsorption, that you're not absorbing food and various other antigens properly, and we'll go into that in a moment. And of course, an increased exposure to toxins and antigens which normally would not be able to get in, but they do, and there is a breach in that wall, in that defense. And that is one of the reasons why all of that lymphoid tissue I told you a moment ago, the gut-associated lymphoid tissue, also known lovingly as GALT, is a containment system. It sits there, that immune system sits there, and together with the good bacteria, the commensal bacteria that sit in our gut, in the gut lumen, is on a moment-by-moment basis preventing potentially harmful intestinal antigens from getting into our systemic circulation. And so it wants to keep the tolerance that we have quite high, so that we don't end up having an immune response to stuff that we really shouldn't be responding to. And I want you to really look at this thing, IgA secretion, because we're going to be discussing this at the end with some very, very important information about that. So when you talk about intestinal permeability, what we're really talking about is movement across that gut wall. And we do know that antigens get across the gut wall in two ways. One of them is that it gets through between the gut cells in a paracellular diffusion, and it also goes right through the cell in a transcellular transport. And we're going to discuss what that means in a moment. But what is this thing called zonulin? Well, zonulin was discovered by Alessio Fasano and his group in Maryland about 20 years ago. And it was their work on an enterotoxin, a toxin that's produced by the bacterium that creates or forms cholera, that terrible disease. And they found that it is able to regulate the junctions, the tight junctions between the cells of the gut. It is a one-layer barrier, just one layer, and it is able to control what gets in. And when people have cholera, they end up having tremendous problems, because those tight junctions, the barrier that sits between those cells that normally keeps the barrier closed, will suddenly open. And when it does, not good things happen. So they were able to find a human analog to this toxin, and they called it zonulin. It is actually a pre-haptoglobin 2 molecule that is found on chromosome 16, and it is an extremely important entity, because we know that the physiologic role of zonulin is that it controls the opening of those tight junctions between the cells. And so really it is a defensive mechanism. It is really there to flush out microorganisms. If a bacterium gets in, the body says, get that bacteria out of here. And so what it does is it puts it in some liquid, and it says, push it back into the gut. But to push it back in, you have to open up those tight junctions. And so the tight junctions is a door that has stuff going both in and out of the body. And that is what really is the important part. And what we were able to find is that zonulin is not only able to control those tight junctions, but it is able to do it in a very rapid way, in a reversible way, and in a very reproducible way. So that means that we can have a biomarker of what is going on within the gut. So we know now that zonulin, to date, is the only physiological modulator of this inter or paracellular tight junctions. And it helps control macromolecule movement, because the body and the cells will not normally allow macromolecules in. They'll allow very tiny or micro molecules of food or whatever it is to get in, but not macro. So when they're getting in, that's not good. And the body responds to that in a very, very integrated way. So these paracellular tight junctions are now known not to be static. We don't have a brick and mortar closure between cells. It's actually quite dynamic, and it adapts to various types of developmental, physiological, pathological, and various dietary circumstances. And what it does is it's a defense against infectious agents to keep them out, or to send them back out if they manage to get in, to absorb nutrients, at the same time to be a surveillance to make sure that gut bacteria don't get in where they're not supposed to, and to transport the infection fighters, the leukocytes, into the gut when necessary. And we call that DAST, D-A-S-T. Very important. And here we have a picture of all of the different proteins that make up these tight junctions between the cells. Actually, very elegant. So what does that mean for us? Well, it's now known that hyperpermeability or increased permeability is extremely important in autoimmunity. And it comes from what we know as a hygiene hypothesis. We know that in the last 60, 70 years, we have decreased infectious diseases that you see on the right dramatically. So we don't have as much rheumatic fever, measles, mumps, you name it. That's gone down dramatically. But what has gone up at the same time are various kinds of autoimmune problems, even epidemics, whether it's asthma, multiple sclerosis, diabetes, Crohn's disease, the list is endless. And one of the reasons for that is that with our emphasis on hygiene, with our emphasis on decreasing diversity of the bacteria that we have, we are now prone, at least in industrialized countries, to now being susceptible to a lot more autoimmune problems. And so those diseases are legion, but we're going to concentrate on just a few, and they're mostly the inflammatory bowel diseases like Crohn's, ulcerative colitis. Those are actual inflammatory diseases of the gut, celiac disease, diabetes, etc. We're going to concentrate on just one of the many types of diseases that one can have, or other types of entities. And we're going to look at celiac disease as a predicate, because that's where the work on zonulin was done originally. And we know that celiac disease has become almost epidemic in the last 60, 70 years. And there's a real question as to why that has happened. Well, if we look at the way we describe pathogenesis of inflammatory disease, let's look at it when it comes to celiac. With celiac, there's a genetic predisposition. 95% of people with celiacs have receptors which make it more prone to being able to have that disease. These are the HLA, DQ2 or 8 receptors that make those people much more prone to it. There is an environmental trigger. In this case, it's gluten, or to be more particular, a component of gluten called alpha-gliadin. And the dysbiotic behavior that takes place in the microbiome because of that, and of course, the increased intestinal permeability. Turns out that celiacs have receptors on their cells, which are shown up here in genetic predisposition, that makes them create anywhere up to 20 times the amount of zonulin of people who are not celiacs, and that therefore opens up their tight junctions much, much greater, allowing all kinds of bad stuff to come into the system. So gliadin in the diet is one of the causes. And why is it a cause? Because the body perceives gliadin in these people as a pathogenic organism in the same way as if it were a bacterium that is pathogenic and it wants to get rid of it. And that's the same as it would with all kinds of dysbiotic growth, whether it's increased bacteria, fungi, or various other kinds of parasitic infections. So when you look at the factors that are driving the rise in autoimmune disease, we now have a pretty good handle on what is causing it. And we're not going to discuss a lot of these in detail. For that, I can have another lecture with you at any time that you'd like. But normally, people who have been vaginally delivered, who have proper nutrition, they've had very low infection numbers when they were young, and they've had very little antibiotic therapy when they were young, especially in the first 2 or 3 years, are going to have a microbiome composition that is going to lead to balance of the bacteria in the gut. Whereas those people who had C-sections, improper nutrition, a lot of multiple infections in their first 2 or 3 years, and with that, a lot of antibiotic therapy, they are going to have being much more prone to dysbiotic bacterial conditions in the lumen. So for those with balanced microbiome, their lymphoid system in the area of the gut is going to have a high threshold. It is not going to respond very easily to problems. And because of a genetic predisposition with that, they are normally going to respond in a way in which they are going to have stable health. But those who had dysbiotic behavior, they have an inappropriate immune response, very low threshold, and they're going to have a tremendous immune response to stuff which they shouldn't have, and therefore they are much more prone to very pro-inflammatory responses to antigens, and they are extremely prone to chronic diseases and, of course, to various kinds of epidemics. So let's take a look at a second thing, which is histamine and diamine oxidase. I'm just going to give you a quick précis. Histamine has been known for a very long time. It's a biogenic amine. It is an entity that is involved in many things, local immune responses, a lot of physiological functioning in the gut, as well as a neurotransmitter. And we're going to really concentrate on its regulation of function in the gut. Histamine is produced both exogenously and endogenously, meaning it comes in with food and bacteria, and it is also produced by various kinds of immune cells in the body. It is also broken down by an enzyme in the gut called diamine oxidase, and it is stored in little cell plasma membrane vesicles of mastocytes, which are sitting right in the gut, and it is secreted upon stimulation. So it is able to scavenge up histamine that comes in when it's ingested with histamine-rich foods, various wines, beers, fish, you name it, various vegetables and fruit. And if you have decreased DAO, which is what we're going to call diamine oxidase, if there's a decrease in that activity, it can be an indicator of intestinal mucosal damage. So let's look at this busy slide and tell you that here is the gut wall, this single layered gut wall, and inside the lumen, you've got bacteria and various kinds of food antigens. The bad stuff is being looked at by secretory IgA in a little mucus layer of the intestinal gut. But if zonulin opens up the tight junctions, some of these antigens get in. And when they get in, they create an immune response, which is regulated by lymphocytes, but you can get an overimmune response, which causes a lot of cell recruitment and an important thing, which is the reactive oxygen species. And we're going to talk about that in a little bit more in a few minutes. But one of the other things that we know is that the immune response has what's called mastocytes. These cells are sitting around waiting to see what happens if there is an immune response required. And if you have stress, and there's a lot of cortisol that is emitted, that is going to stimulate the mastocytes. And what do they let go? They let go of a number of stimulating proteins, one of them being histamine. And so that can create an inflammatory response. So the histamine effect, we now know, is well understood. Within the plasma, we know that there's normally about 0.3 to 1 nanogram per mil. That is considered a normal range. Anything above that will begin to cause issues. And so we know that there's very little issue between 0 and 1 nanogram. If you get between 1 and 2, you start to get increased gastric and intestinal secretions, and also some increase in heart rate. You don't want to see histamine going above 2 nanograms per mil. Just to let you know, as you can see, it can go up dramatically, leading to any number of problems, all the way up to bronchospasm and even cardiac arrest. God forbid you run into any of those problems. And in the reports that we give to our health professionals, we do show you all of those levels, which I'm not going to spend time on now, but we've got these beautiful, colorful reports that will explain the various levels of these entities. Let's look at the microbiome and the gut, and look at something about food sensitivity and Candida, which is a yeast. And in order to understand that, we do need to know something about this inner bacterial ecosystem that we lovingly call the microbiome. The cells and the bacteria in the intestinal area and the rest of our body, on our skin and other areas, outnumber our human cells 10 to 1, as I mentioned earlier, 10 to 1 or even more. And the genetic material in these cells, the total genetic material, outnumbers the human genetic material by 150 to 1. So there are well over 3 million bacterial genetic codes versus, about 25,000 human genes, which we now know exist after the Genome Project. And we know that there are at least, and probably a lot more, than 1,000 bacterial species in the body. And when you have evacuation of the colonic content, when we have poo coming out of our system, fully 50% or more of that is bacteria, the excremation of bacteria out of our system. So it represents a tremendous amount of the feces that are in our gut. So we're going to really just quickly go over a couple of things that I think are important. And for those of you who would like to know more about the microbiome, we can have a separate lecture on that. But let's just take a look very quickly at something that is of interest. When you look at breast milk, for example, maternal milk, of the many things that are in it is something called human milk oligosaccharides, the HMOs. And so that represents an important part of maternal milk. It's unique to human breast milk, despite the fact that it's not digestible by the infant. The infant cannot digest human milk oligosaccharides, and yet it is a third most abundant component of human milk after lactose, fat, etc. So why is it there? Well, the answer is actually very interesting. It's there because it functions as a prebiotic. It's there to help nourish and establish the commensal bacteria in the newborn. So that's fascinating. So what it's saying is, right from the get-go, the system knows that the bacteria need to be there. They're critical to proper functioning of the newborn and the young child. And so it's giving them the food, the bacteria, the food that they need to be able to function properly. And when we do look at this, we know how essential the bacteria are. If you look at the upper right, you're going to see that when we ingest carbohydrates, we ingest it as monosaccharides, polysaccharides, and the body is able to absorb simple sugars very, very quickly. It absorbs them through our cells, but it cannot absorb polysaccharides, the larger saccharides. So what happens? When you go from the proximal to the distal small intestine and then into the colon, what happens? It is the bacteria that are beginning to work on these polysaccharides, to ferment them and to turn them into short chain fatty acids, which our body can absorb. And so we know that humans only have about 20 genes that will encode for proteins that can break down carbohydrates. Bacteroides theta alone, and it's one of 1,000 or more species, that bacterium alone has 260 genes that will break down those polysaccharides. So it's not an overstatement to say that the microbiota are actually essential for digestive function, not just something we'd like, they are essential. And so when you look at an alteration in the gut microbiota, what are we looking at? Well, remember, I spoke earlier about the environmental triggers, and we talk about chemicals, infections, the dietary proteins. That will lead to change in the microbiota, and it can lead to either eubiosis, meaning a normal functioning microbiome, or it can lead to a breakdown in that normality. And that group of microbiota interact with the host to either allow for a proper hemostatic system, a well-balanced immune system, which will lead to healthy, stable state, or just the opposite. It can lead to systemic inflammation, which can lead to a whole number of things. And I'm just giving you a very small list of asthma, arthritis, multiple sclerosis, obesity, which is an epidemic in North America, metabolic syndrome, and even brain disorders. Keep this in mind. We're going to discuss it in a few moments. And food sensitivity, we know about 17%. We have a pretty good idea of what the causes are. Just 17%. The other 83%, dysbiosis, a improperly functioning gut bacterial world in which you get a change in the percent of commensal or good bacteria to the pathogenic, or what we'll call bad bacteria. If those good bacteria go down to less than 85%, the problems begin. And so it's extremely important to be aware of what the microbiota are doing. And when we do our tests of total IgG food sensitivity, what we're looking at is to get an idea of what the various foods are doing to the enzymatic balance within the gut. If that becomes imbalanced, either due to protease problems from the stomach, pancreatic origin enzymes, or bile, that can throw the system out. And that will allow for yeast to begin to grow or to even overgrow. And you get into the ubiquitous problem of candidiasis. And so one of the other tests that we offer is a Candida suite. And we now offer only the Candida suite, not just Candida IgG. We really want to know what these three antibody groups are doing. It is an antibody screening test for proliferation of Candida, and it lets us know what are these three immunoglobulin groups doing. Is IgM up? Because that's going to tell us if you're in the beginning of a yeast infection. Is it IgG? That develops later, and it's going to tell you if you had a past or even a prolonged infection. Or is it IgA, which we know is present almost exclusively in the mucous membranes? When it gets into blood, that's not a good sign, because it indicates that there is an ongoing frank infection that is taking place. And one of the things that causes it is stress. You get an increase in stress, you get a hypersecretion of cortisol by the adrenals. And that in turn is going to compromise digestion and absorption, the way I described earlier. That's going to lead to inflammation of the intestinal mucosa, which then leads to more production of cortisol. And of course, that creates more stress. So that is what's known as the stress-induced hyperpermeability cycle. Keep this in mind, it's extremely important. And I want to then tell you that from that, you get a complementary assessment. All of the tests that we're doing are screening tests. These are not diagnostic tests. They are specifically not diagnostic, but they are extremely important. What is the primary screening test? I'll submit to you that it is serum zonulin, and I'm giving it as a 5-star rating. The only ones we're talking here are the 5-star ratings. There are 4-star, 3-star. There are many tests that you can do. I'm only looking at the really important ones here. So this is the key. This is the foundational test. No matter what else you're going to do, I would humbly suggest that this should be added to your testing sequence. Activation of the immune response. You want to look at the immunoglobulins, especially immunoglobulin G for food sensitivity, total IgG. That's going to identify the antibody antigen complexes, mostly of food, that are causing inflammation in the gut. Added to that, the Candida. And by the way, we're talking about Candida albicans. I should have said that. There are different types of Candida, but Candida albicans represents somewhere between 80 and 90% of the yeast types that are in the gut. So you want to know, is there an overgrowth of this particular common yeast? Because it is a classic manifestation of dysbiosis. Thirdly, allergic inflammation and the ability to decrease inflammation. What are we talking about? We're talking about histamine that I mentioned earlier and its breakdown enzyme DAO. It's not only important because histamine is so ubiquitous, it's also important because DAO in many people is quite low. And the good news is that with diet and or supplements, you can recalibrate the amount of DAO and therefore control histamine. And last but not least in this group of 5-star rated tests is the test for stress. And that is extremely important when it comes to permeability and absorption. And I didn't let you know earlier, but it was mentioned by Jennifer that we also do salivary hormone tests. You can pick them up from us or others, but it's extremely important. If you do nothing else, then at least look at a diurnal cortisol and DHEA, because it's going to tell you how well the adrenals are functioning, and it's going to tell you how well the body is able to deal with the stresses in life, which we're not discussing in detail in this particular hour. Hot off the press, I wanted to give you an idea of how important the gut is. This is information from yesterday, and it's from an article that just came out in Nature by Fitzpatrick and his group, and it's called Gut-Educated IgA Plasma Cells Defend the Meningeal Venous Sinuses. Now, what does it mean? Well, we do know that IgA, secretory IgA, is an immunoglobulin, and we know it well because it sits in the mucus layer in the gut. That's where it picks up a lot of the antigens from food and bacteria, extremely important, like Candida. We now know that it sits not just in the gut, but it also sits in the brain's dura mater or dura mater, in that lining, and it's a barrier against germs. So lo and behold, we know that both the brain and the gut contain IgA secreting plasma cells. And they, interestingly enough, the ones in the brain, where are they acquiring their ability? They're acquiring their ability from the plasma cells that have been trained and are getting their capability originally by dealing with gut bacteria in the intestine. And we now know that. And this article, which I read by Fitzpatrick and his group, is very elegant. With DNA sequencing that they did from IgA cells in the gut, they were able to relate them with sequencing to the meninges in the central nervous system. So what does it tell us? It tells us that gut educated antibody producing cells like this IgA inhabit and defend regions surrounding the central nervous system. But where are they getting their training? They're getting their training in the gut. So lo and behold, the gut-brain axis is alive and well and extremely important. And with that very short talk, I want to thank you, and I am open for questions.